Okay, the next structure found in prokaryotic cells or bacteria is the plasma membrane. So the plasma membrane acts as a barrier to separate the cell from the environment. Okay, so right here, separates the cell from its environment. A quick review of the cell membrane and how it works. Remember we talked about phospholipids being hydrophilic and hydrophobic, so the heads are repelling water and the tails are, the heads are attracting water and the tails are repelling water. So maybe we'll add that in so you don't forget to write that down. So hydrophilic means it likes water. Sorry, I'm trying to write with my finger today, so I'm not doing a great job. And hydrophobic means, remember, it fears water. All right, and it's a double membrane, so all of the heads that are up here on the top are interacting with water, and then all the heads that are down here at the bottom are also interacting with water. Where the water can't get through, just through the phospholipids on its own, is this middle layer of the sandwich right here, where all those tails are, those hydrophobic tails. So that's what's repelling the water. This, in the end, means that the cell membrane is semi-permeable. So some things can go into and out of the cell, but other things can't. So it really is able to select the items that are coming into and out of the cell. We'll talk a lot more about the plasma membrane in a later podcast. Okay, prokaryotic cells like bacteria also have what's called a cell wall. You may have thought before that only plants had cell walls, but bacteria also do. Their cell walls are made out of something called peptidoglycan. Highlight that for you. Right here, peptidoglycan. It is a polymer. We talked about polymers last unit. And basically, it's chains of these peptidoglycan monomers, which are basically made of glucose, kind of a version of glucose, two different versions of glucose. So what I really want you to know is they have these cell walls. The cell walls are made of peptidoglycan, and what it does is down here in the corner, it gives real rigid support to the cell itself, so it helps support and structure the cell. But it does allow solutes, so particles, to move in and out of the cell. So it's not like it's a barrier. It's not completely blocking things off. It does allow molecules the cell needs to come into the cell and things the cell wants to send out to go out. In this diagram that I'm showing here, the peptidoglycan would be this area around the edge here. And then the red layer that's in there already is the cell membrane, or the plasma membrane. Okay, another part of the cell wall is those granules that we talked about earlier. So outside of the peptidoglycan layer is another substance, a type of granule, that we use to actually identify bacteria. And the granules can either be gram-positive or gram-negative, they're called. Gram, I think, actually refers to a scientist who discovered this. That was his last name. So either they have the granules or they don't. And there are two pictures of those, what those bacteria might look like. They're stained. So gram-positive, as you can see, stain that kind of purplish color, whereas gram-negative are kind of a reddish color. And that's what we do. We take bacteria, we put this gram stain on them, and that will tell us what kind of granules they have in there. So the real important part of this, again, if you're writing this in your notes, is just that we have two major groups of bacteria whose granules stain differently. So we've got the gram-positive granules and the gram-negative granules. Why do we care about that? Well, what happens is, in this gram-negative cell wall, okay, which we see right here, there's this extra substance that things like drugs like penicillin can't get through. And so depending on what type of bacteria it is, if it's a gram-negative bacteria or a gram-positive bacteria, we might treat it differently, might use a different type of medication to help someone out who is infected with that type of bacteria. So being gram-negative or gram-positive as a bacteria is helpful knowledge for us as we try to treat different bacterial infections. Okay, another structure found in prokaryotes, or something that prokaryotic cells can actually do, is create something called endospores. And what these are like little structures, little capsules basically, of when the bacteria goes dormant, stops growing, stops reproducing, stops most of its function, but then can kind of reawake after a time. So what happens is when conditions are really harsh and the bacteria don't have the right types of conditions to survive, so maybe it's really cold or it's really hot, or they're being exposed to dangerous UV light, or they don't have enough nutrients, they'll form these endospores, these little coverings over the top of the bacterial cells, so that they can just survive, basically. Like I said, they go dormant. And then once the conditions come back to normal, then they'll start reproducing and dividing again. And so this is something that's very common, for example, with the anthrax bacteria. Anthrax, you've maybe heard, is a bacteria that's been considered as use in like military grade weapons and things like that. And so what they're actually working with is the endospores of the anthrax. That's the white powder that you maybe have heard of with anthrax. And then when anthrax gets into its good conditions, whatever that might be, inside someone's respiratory system, for example, then they kind of wake up, the bacteria are no longer dormant, and they start reproducing and causing problems. All right, prokaryotes do have some things that stick out of their cell walls as well, these surface appendages, that allow them to do lots of different things. For the most part, they are made of proteins, these surface appendages are. So the first one is the flagella, which the picture of is right up here. So the flagella allow for movement. Not all bacteria have them, but especially those that live in water, aquatic environments, flagella helps them to swim around. Another type of appendage is the pili, or if you call one of them a pilus. They are tubes that come out of the bacteria that are shorter than flagella, but they can also be used for movement, like grappling hooks. So like the bacteria will toss out a pili or a pilus and try to attach to something and pull. Also can be used for conjugation to transfer plasmids. Now we'll talk more about this a lot later on, but basically two bacteria can hook up and exchange DNA with each other. And the pilus is this tube that allows them to transfer DNA from one bacteria to another.